Today we're going to be talking about the cell cycle. And the cell cycle is a timeline. It's the time from when the cell is born until it divides. The cell cycle can be illustrated by a circle. And we're going to divide this circle into 4 stages: G1, S, G2, and M phase. Let's go ahead and get started down here. This is where the cell is going to be born. The first stage of the cell cycle is going to be G1. So we'll write G1, and that stands for gap 1. This is where the cell grows to its adult size, and the cell carries out its function. It does what it's supposed to do. So if it's a neuron, it's going to conduct electrical impulses, meaning it's going to conduct messages. If it's a muscle cell, then it's going to contract and relax. If it's a digestive cell, it might make digestive enzymes. So that's what it means to carry out its business. The cell does what it's supposed to do. The next stage of the cell cycle is going to be S phase. And S phase stands for synthesis. What we're synthesizing here is DNA. We're actually going to replicate all the DNA. So copy DNA. And we need to copy the DNA because we're getting ready to divide the cell. And each cell needs its own copy of the DNA, of the information, in order to function properly. This stage also tends to be the longest. Not always, but usually. And that's because quality counts here. We want to do a good job in replicating that DNA. Any mutations puts the cell in danger for not carrying out its business, not being able to function properly. And we also do not want to replicate a defective cell. So quality counts here. And quality takes time. The next stage is going to be G2, which stands for gap 2. In gap 2, the cell is going to grow a little bit larger. And the cell is also going to make the necessary proteins for the last stage, which is where the cell actively divides in 2. And that requires some specialized proteins. So we're going to go ahead and we're going to make those proteins ahead of time so that we don't have any problems when we do get into that last stage. So cell makes proteins needed for cell division. Lastly, we have the stage where the cell is actively dividing. This stage is going to be called M phase. M for the mitotic phase. We can divide M phase into 2 smaller stages. The first stage is nuclear division. And there's 2 ways to divide the nucleus here: mitosis or meiosis. Now mitosis, we're going to make an exact duplicate of the DNA. And we're going to use that for somatic cells or body cells. In meiosis, we're going to change up the DNA a little bit. We're going to make some changes on purpose. And this is going to be used for reproductive cells because we don't want to have kids that look exactly like us. In terms of evolution, and evolution means a change over time, is the Earth changes over time, species need to change too. So we want cells that are a little bit different from the parent cell. And that way they can change over time and keep pace with environmental changes. So both of these, however, are going to be concerned with nuclear division, or division of the nucleus. The last stage over here, which is also part of M phase, is going to be cytokinesis. This is where we actually divide the remainder of the cell. And as you can see right here, these two phases overlap. Cytokinesis is going to begin before mitosis or meiosis are completed. So there's an overlap in time. Now if we go back up to G1, the cells are actually capable of leaving the cell cycle if they want. And they go into a special stage called GO. This is a non-dividing stage. In fact, most of the cells of the body ultimately will go to GO. And in GO, all the cells are going to do is carry out their function. Now we've got two populations of cells in GO, this non-dividing phase. One population of cells, once they get to GO, can't leave. That's it. Once they go to GO, they stay in GO and they cannot re-enter the cell cycle and divide again. That population of cells would be, for example, neurons or muscle cells. We don't want to injure these cells because when we injure or kill off these cells, that's it. We cannot make new muscle cells. We cannot make new neurons. That's it. Whatever is damaged is gone forever. The other population of cells that are in GO are capable of re-entering the cell cycle and dividing if they receive the proper signal. For example, a hepatocyte, which is a liver cell. If part of the liver becomes damaged, part of the cells die, the healthy cells nearby with the proper signal can re-enter the cell cycle and begin to reproduce and replace those dead cells with healthy cells. And then once they've replaced all those dead cells, they again exit the cell cycle and begin to just simply carry out their business in GO. Here we have G1, S, and G2. All three of these stages together can be called interphase. And interphase is just that. Inter means between. So this is the stage or phase between M phase, between when the cell is actively dividing. Interphase consists of G1, S, and G2. Also, between these different stages of the cell cycle, we have checkpoints. For example, our first checkpoint is going to be right here. And these checkpoints tend to be towards the end of a particular stage. So since this checkpoint is at the end of G1, we're going to call it the G1 checkpoint. A checkpoint is also called a restriction point. And what we're going to do at this checkpoint is we're going to check to make sure that the cell has attained its proper size and that it's carrying out its function properly. If not, the cell is not going to move into the next phase because we don't want to replicate a defective cell. So at that point, the cell will try to repair itself, and if it does that successfully, it will then move on to the next phase. Our next checkpoint is going to be at the end of S phase. This is going to be our S checkpoint, CP for checkpoint. The S checkpoint, what we're going to do is to make sure that that DNA that we copied is accurate, that there's no mutations. If this phase checks out, then the cell will then move on to the next phase. And towards the end of G2, we're going to have our next checkpoint called the G2 checkpoint. What we're going to do in the G2 checkpoint is to make sure that the cell is the proper size, and we're also going to make sure that the cell has made all the proteins necessary for cell division, because we do not want to get to cell division and find out that we don't have the necessary materials to complete cell division. So before we move on, we're going to make sure that all those necessary materials, proteins, are ready to go for cell division. Then we're going to get into M phase. And our last checkpoint is going to be right in the middle of M phase. Right here. And it's going to be called M checkpoint. M for metaphase checkpoint. What we're going to do there is we're going to make sure that the chromosomes are lined up in the center of the cell, and we're also going to make sure that the spindle fibers are going to be firmly attached to the chromosomes, so that when we separate the chromosomes, and half are going to go to one cell and half are going to go to the other cell, we want to make sure that those spindle fibers pull half of the chromosomes to one cell and half to the other. If there's a mistake there and the cell receives an extra chromosome and the other cell receives is missing one chromosome, then both cells, that's going to be a lethal mutation. That's going to result in cell death of both cells, and we don't want that to happen. So we're going to check to make sure that those spindle fibers are firmly attached before proceeding. And that's going to be it for the cell cycle.